I think that was a really wonderful talk, and I personally think that the way we've divided oncology into looking at various tissues is kind of an accident of history, because light microscope was invented in 1590, and but looking at tumors as a colon cancer, a breast cancer, a lung cancer doesn't really tell us anything about the underlying alterations that are driving that cancer. So we really didn't have next generation sequencing until the human genome was sequenced about 400 years after the light microscope was invented, and it was really 2012 where it became clinical grade. We could do next generation sequencing, but again, I don't think we need to follow the paradigm just because that is 400 years old. I think we really need to rethink it. And that is the essential part of moving oncology ahead. Clearly, we were not doing very well in oncology, just looking at tumor of origin for patients. And the reason was because we were really not understanding what was driving each patient's tumor. So what I'm going to talk about is what I think is now the next generation, and that's N-of-1 patient-centric clinical trials, and the fact that cancer is like the nine-headed hydra. It has many drivers. Each individual cancer has many drivers. And really, to kill the hydra, you can't just cut off one head, because like the mythical hydra, if you cut off a head, it grows back two heads. You really have to, in order to kill the hydra, you have to cut off all its heads. And really, that's what we need to do in cancer. And I think really that's the next generation of trials. So these are my disclosures. And just a little bit of background about myself. Before my current position at Medical College of Wisconsin, I was founder and director of the Center for Personalized Cancer Therapy at University of California, San Diego Moores. And before that, I was the founder and chair of the Department of Investigational Cancer Therapeutics, which is the phase 1 department at MD Anderson Cancer Center. Probably became the largest academic phase 1 clinical trials department of its type, maybe anywhere in the world. And I mention this because these experiences in these two different institutions really informed my thinking about cancer and where we need to go. So I think the next generation moves us, I hope, from drug-centric clinical trials. So clinical trials and clinical practice especially clinical trials, put drugs at the center of the universe. And then we find patients that fit onto the drugs. But really what we need to go to is patient-centric clinical trials, where the patient is the center of the universe, and we find drugs to fit the patient rather than patients to fit the drugs. But that really needs a major rethinking of how we are treating patients and how we're doing clinical trials. Now, the Target Cancer Foundation's TRACK trial, I think, is a great example of a patient-centered trial. Each patient is recommended an individualized therapy, and so it's an N-of-1 trial. Each patient is specifically for rare cancers, but the idea is really to look at every cancer as a rare cancer, not just rare, but an N-of-1, and to try to get the best therapy for each patient. The TRACK Foundation trial also has the advantage of being what I call a home run trial. Patients can stay at home and get their therapy while at home, so it's a fully remote trial. So take-home points that I'm going to try to get across is this is about getting the right drugs to the right patient at the right time. And what does that mean? At the multi-omic level, every metastatic tumor is unique and complicated, just like malignant snowflakes. Each snowflake has a complex crystal structure and is unique. Metastatic tumors are the same too. And that means that metastatic disease may require require individualized, customized combination therapy. That is not what we have done in the past. We try to put patients in buckets and give them all the same therapy. And then the pillars of precision medicine are genomics and immunotherapy, and they are not separate, as Dr. Savaya mentioned. They are a couple, and they are really married to each other. So we have now really powerful technology and bioinformatics that can do amazing things, including, and I want to mention this, discovery in real time. So for most patients in the past, if you had to wait for a clinical trial, if you had metastatic disease, you would definitely, the patient unfortunately would die way before you had the clinical trial, because it takes months and years to develop a lot of these clinical trials. But I want to give you an example of what the power is of doing genomics and transcriptomics and discovery in real time. So this is a fibrolamellar cancer patient. The patient has what's called an undruggable fusion, a DNA JB1 PRK-ACA fusion. We know fusions are very strong drivers. But together with our colleagues, we exploited multi-omics. We looked at transcriptomics and bioinformatics to explore synthetic lethality for a single patient. And this was a young woman, about 30 years old, and she'd had the cancer for a while. She'd had various therapies and actually had been told to go to hospice. And the bioinformatics analysis came up with a non-intuitive therapy. So she has this undruggable fusion. She'd already had a type of immunotherapy called atezolizumab, which is PD-L1, an anti-PD-L1, and she had not responded to that. She had low tumor mutation burden and was PD-L1 negative, both of those being markers for immunotherapy. So it was completely non-intuitive, but the analysis came up with her possibly responding to a combination of two immunotherapies, both of which are approved in multiple countries. And she got those two immunotherapies, and she went into complete remission. And the time that we published her, she was in complete remission. She's on the internet, so I'm not violating her privacy. So she was 2 years in complete remission. She's now 4 years in complete remission, and I think she's probably cured of her disease. So I'm not saying that we can now do this for everybody, but I am saying that we can now begin to see the glimmer of the potential of what we can do and really do in real time for patients. Okay, so let's go to the reclassification of cancer. In 2014 and 2015, we published a paper suggesting that molecular aberrations do not segregate well by organ of origin. And we looked at particular HER2/3+, we looked at it in almost 38,000 patients. And at the time, the only patients that were getting HER2-directed therapy were breast and gastric cancers. But we found that HER2/3+ was found in multiple different tumor types, like bladder cancers, cholangiocarcinomas, very common as salivary duct tumors, actually. So now we have an FDA-approved therapy that is tissue agnostic. And but it took 10 years from the time that it was known that this was a target across cancers to get this tissue-agnostic therapy. So N-HER2, trastuzumab deruxtecan, it's an antibody-drug conjugate. My experience with it is that it's a really good therapy. And now it's available for any patient that has HER2/3+, including any patient with a rare cancer. So I think that's a tremendous advantage. Okay, so evolution of clinical trial design and what has happened, and then what is the future? Because I think The tumor-agnostic therapies are just the beginning, and so I want to address that a little bit. So we've gone from redesigning cancer clinical trials. We considered lung cancer as one disease, and now we consider lung cancer as multiple different diseases with molecular subsets, EGFR, BRAF, and so forth. And then the next revolution was the tissue-agnostic therapies, where we looked at, let's say, the examples that Dr. Sabaya gave, and that has led some of these really important trials with BRAF or RET fusions, finding that they're important in multiple different tumor types and getting an approval across tumor types. And so this has led us from large randomized studies where just of unselected patients, where very small numbers of patients respond, to very small studies, anywhere from 30 to 100 patients. And you do the work up front to find the right patients, but a lot of those patients respond. They have very high response rates. And I'm not going to go over this too much, but we now have multiple tumor-agnostic approvals. And I think that the potential is for virtually any targeted drug to be a tissue-agnostic approval. But I think there's more, and this is really important. We have to think about what is the next generation and what is genomics really telling us. And when we started doing next generation sequencing on our patients, we began to realize that every tumor is very complicated and every tumor is distinct. So this is some sequencing on some breast cancer patients. I've overseen probably about 21,000 patients that have been sequenced, but I want to give it as an example because I think it's a visual example. So both of these tumors have ERBB2 amplification, which is HER2, so they can be targeted by HER2 targeting agents. But if you look at the patients, they have complicated molecular portfolios, and the rest of their aberrations are different from one patient to the other. And I think it would be naive to think that just targeting one aberration would be enough to really make important headway in these patients. And furthermore, I think it would be naive to think that these two patients need identical therapy the way we've been doing it. These patients both need an individualized, customized therapy regimen that targets all the drivers that they have. So this brings a new clinical trial design that uses multi-omic interrogation, not just DNA, but RNA, immunomics, and so forth, and personalized combination therapies. And we did an initial trial of this type that we called Predict for profile-related evidence determining individualized cancer therapy, that we started in 2015 at the University of California, San Diego. And the idea was to individualize drugs for each patient. This trial was a master protocol. It allowed any patient on the trial, and it treated each patient as an N-of-1. So it's a combination of what we call a basket trial, an umbrella trial. Each patient was a molecular tumor board that advised on therapy, and then we tried to match the patients with a customized combination of drugs. And this was published in Nature Medicine, and what we were able to show is for these highly refractory metastatic tumors, if patients were well matched, their response rate was much higher than if they were poorly matched. And even though they were heavily pretreated, quite refractory patients, for the well-matched patients, their overall survival at 2 years was over 60%. And for the poorly matched patients, their overall survival at 2 years was under 20%. And again, this was published in Nature Medicine and did get a little bit of attention. It was featured in the Future of Medicine, National Geographic, and the NIH Director's Blog. And I was quite honored to ask to present at the Nobel Symposium for Precision Medicine last year. So just to give you an idea of how patients were treated. An example of a patient. This is a 62-year-old man with poorly differentiated cancer of unknown primary. So this is a rare cancer, and it's a cancer, it's almost ideal for molecular interrogation, although I believe molecular interrogation should be done for everybody. But in this case, we don't actually know the tumor of origin. So this patient had a KRAS mutation, which some can be targeted by MEK inhibitors. So we gave the patient a MEK inhibitor called trametinib, and the patient had an ARID1A alteration, and there's evidence that these can be targeted by PARP inhibitors. So we combined the trametinib with a PARP inhibitor called olaparib, and this is the patient's response. You can see that the patient had a really excellent response, and this was ongoing at 2 years. So there's some questions that naturally come up because this is really kind of shattering the normal paradigm in many ways of how we treat patients. So the first question that is often asked is, if every patient gets a different drug regimen, what is the endpoint? How do you figure out if this is effective or not? And the answer is you analyze the robustness of the algorithm determining how you match patients, not the individual drug combinations, because you're never, if every patient gets an individualized combination, you're not going to be able to analyze individual drug combinations, but you can analyze the algorithm. And the second problem that comes up, which I'm very familiar with, is because I ran the phase 1 department at MD Anderson Cancer Center, which is the listed as the #1 cancer center in the country. So the problem is dosing. How do you dose these patients? And so let me share how we now decide dosing. And so the current phase 1 trial paradigm for dose finding is, the classic way we do things in phase 1 is we have what's called a 3+3 design, and you have cohorts of 3 patients per dose level. And in the first cycle of therapy, you determine how they did. And then you can also have an accelerated design with 1 patient per dose level. And the maximum tolerated dose is determined sometimes on 6 patients. Now, of course, we do expansions, but it's not a lot of patients. And phase 1 trials take at least 1 to 2 years to complete. So you can see some problems here. Dosing is determined on small numbers of patients, and this takes a long time to do. And so why is this a problem? So if the majority of metastatic cancers are unique and complicated, and each patient needs a tailored treatment regimen, if there are 300 drugs in oncology, the number of 2-drug regimens is 45,000, and the number of 3-drug regimens is 4.5 million. And furthermore, if the average tumor has 5 molecular alterations, there are over 1 trillion patterns. So that's the number of patterns. So if we're going to do phase 1 trials for all of this, it'll take over 1,000 years to figure this out, and we need a different paradigm. This doesn't work. There's also another problem with it. As we were doing these novel types of studies that we discovered that, and this kind of makes sense, right? We give a standard dose to everybody. And should these 2 patients receive the same dose? The 20-year-old muscular guy who happened to get cancer, I pulled him off the internet, but I think he actually has cancer. And the frail 80-year-old, I don't think you need to be a physician to know that these 2 patients should not get the same dose. And what dose does she get? Yeah. What we started to do with Dr. Nakunjam, who is here, is we started to analyze patients in the literature. And we did several papers analyzing about 75,000 patients who had gotten clinical trials in the literature. And we came up with a framework for personalized dosing. And the framework gave us an idea of what kind of drugs you can combine, what kind of drugs you need to dose reduce. But the general solution is precision medicine should be about not just individualized drugs, but individualized dosing. And so personalized dosing, instead of interpatient dose escalation with different groups getting different doses, we individualize the dose for each patient. We start with a lower dose, and then we raise the dose to tolerance. And the bonus benefit is that this is probably actually good for drug regimens that have approved dosing as well, because as I mentioned, the 80-year-old and the 20-year-old really should not be getting the same dose. So in the published studies, we found that patients that were more highly matched actually had less toxicity than the patients that were low matches, and the number of drugs in the regimen was unrelated to the number of toxicities. So this can be done safely. It's something that doctors do in clinic every day outside of oncology. If a patient comes in and they have rheumatoid arthritis and they have an infection, there's no hesitation to give them the right drugs for each of their conditions. But oncology has historically not done that, but I think the future is to individualize for patients. So I want to mention that genomics is just the tip of the iceberg. And we have transcriptomics, RNA, protein, immunomics, and so forth. And as part of the WIN Consortium for Personalized Cancer Therapy. We have a mission for global delivery of precision medicine, but also to do innovative trials. And I think we did the first N-of-1 trial that included transcriptomics, and this was called the Winter Trial. And this was also published in Nature Medicine and showed that using RNA in addition to DNA could add to the percent of patients that were matched to therapy, and that RNA, in addition to DNA, was an important component of matching patients to drugs. So I'm just going to finish there, and really I've been very privileged to work with patients for my entire career. I was just thinking what an honor it's been, and I really hope to see precision oncology continue to change the outlook for patients as quickly as possible. So thank you.